Einen wunderschönen guten Montagabend zusammen. Heute mit einem Experiment. Wir haben den ersten internationalen Gast in der Sendung, Garett Slater. Er hat gerade 2022 promoviert, arbeitet momentan am amerikanischen Bieneninstitut und hat vorher in seiner Ausbildung sich um Ernährung von Bienen und Königinqualität gekümmert. Ähnlich so vom Thema ohne die Ernährung, die wir letzte Woche uns angeschaut haben. Momentan liegt sein Forschungsschwerpunkt aber eher im Bereich der Entwicklung genomischer Werkzeuge für die Imkerei, also bleibt beim Thema Imkerei. Und was mich so als Varroa-Resistenzzüchter besonders interessiert, er kümmert sich auch oder versucht sich um den Themenbereich Selektionsmöglichkeiten gegen Varroa und andere Krankheitserreger auf genomischer Ebene, also quasi diagnostische Tools herauszubekommen, wo steckt das drin. Er hat 2 Jahre als wissenschaftlicher und Techniker an der Bee-Informed Partnership in Minnesota gearbeitet. Das ist im Prinzip ganz viel das, was er heute auch tut. Informationen aus der Wissenschaft an die Praktiker herausgeben. Und er selbst ist auch Praktiker, also anders als ganz viele Wissenschaftler hat er tatsächlich bereits 12 Jahre Erfahrung, 8 Jahre davon in der großen kommerziellen Imkerei. Das heißt, er hat schon mal ein paar Bienenvölker gesehen, wahrscheinlich mehr als die meisten von uns, zumal in großen kommerziellen Imkereien in den USA große Völkerzahlen geführt werden. Okay, that's your startup for you. I didn't give an introduction to the content of your presentation. I just
Sure.
focused on you and what you did, what your current work is, what your background for beekeeping is. And I think the content of the talk I will give to you. Before we start, I will give the audience the background for the structure of this talk.
Okay.
Because usually we intersect questions and presentation, and this time we will collect the questions for me to translate them easily to the back or to the end of the session as a question and answer session. Also nochmal für alle zum Mithören. Wir machen es heute ein bisschen anders von der Struktur. Garett Slater wird den Vortrag en bloc halten. Ihr könnt wie üblich Fragen stellen. Ich werde die mitlesen. Ich werde die für eine Fragen- und Antwort-Session am Ende sammeln. Und ob ihr mir die Fragen auf Englisch oder Deutsch stellt, ich habe dann halt die leidige Pflicht, das auf Englisch zu übersetzen. Okay, Garett. Thank you for meeting with us. I'm very interested in your session. Thank you for having the time. I was very surprised when we first contacted and I asked for your images and for your background information that you actually were willing to do this presentation for us. And so I had to think if we can do this, because not everybody can understand English, especially on the scientific level that's necessary to follow this presentation. So that's a little experiment for us, but I hope it works out, and let's give it a go.
Well, yeah. So first off, thanks for having me. I mean, it's always exciting to do research in the States and then talk about what I'm doing on sort of a global scale. And so I'm really excited to talk about the work I'm doing now and, hopefully be applicable to, the German beekeepers. And, maybe in 5 to 6 years, I'll learn German and I give a talk in German, but we'll see. So, first off, you gave kind of introduction about myself. I really want to stress that, I grew up as a beekeeper, in a large commercial beekeeping family. And my uncle, he actually managed, around 10,000 colonies. And so that's a pretty large commercial operation. And so I've really been around it my entire life. And, one reason I really decided to get into research was because, one, beekeeping is really hard, especially if you're doing that on such a large scale. But also, I really wanted to find sustainable solutions for, the beekeepers that I know personally, and beekeepers that I have witnessed their losses. And so, one thing I've always been interested in was, in the United States we have these queen failures where beekeepers will purchase queens or raise their own queens, and they'll fail within 6 months. And you as beekeepers know this, that a queen should thrive and reproduce and really live and not be replaced for 2 to 4 years. And so if beekeepers are losing queens within 6 months, this is a huge issue because queens are really central to a colony. And so I really focused on queen research during my master's degree at North Dakota State University, and then also a little bit with the Bean Farm Partnership, where I did extensive work with beekeepers across the country. And so this queen thing was always something I've been interested in, and something that we do these surveys in the United States and Canada, and queen issues is always number 2 to 3 behind like Varroa and viruses, and so it's definitely a prevalent issue. But during my PhD at Purdue University in Indiana, kind of central United States, I sort of decided to take a different role and focus on the males or the drones, because we just don't know much about them. We don't know much about their biology, and we don't know if there's any issue with drones and if they could actually be contributing to queen failure and queen quality issues. And so, throughout this talk, I'll be talking about my work during my PhD, which I just finished up. I'm currently at a sort of a government institution where we study honeybee genetics. And so I'll talk about that a little bit more, but we'll be expanding on some of this research. But I'm kind of excited to talk to you today about, drones, drone biology, and what we know and what we still need to learn. But before I begin, I always like to stress, why are drones or males important? So when you think about the structure of a honeybee colony or the social structure of a colony, there's typically, thousands of individuals in a colony. There's usually one queen, which is pictured right here, and the queen is the primary reproductor of the colony. She'll lay, 2,000 to 3,000 eggs per day, and that's her only role. Once she mates, she lays the eggs, and she's the primary reproductor of the colony. The rest of the individuals are the smaller workers. Genetically, they're very similar to queens, but they perform a wide range of tasks non-related to reproduction. So they do nursing behavior, they do feeding behavior, guarding behavior, all these really important tasks when you think about a healthy colony. But then we have in the middle, we have these drones. And during peak colony size, there'll be around 20% of the colony will be made up of these drones. And I think a lot of beekeepers don't realize just how important drones are, because they don't do any tasks like a worker. They don't lay any eggs, but they're extremely important for a lot of different reasons. And so I hope really by the end of the talk that you tell your beekeeper friends or people that are interested in honeybees that drones are important and we should really consider them. Now, why are drones important? The first thing is drones contribute directly to queen quality, and in the United States, they could contribute to queen failure issues. And so a queen will typically, in about 7 to 6 days when she emerges as an adult, she'll go on these multiple mating flights, and she'll mate with 15 to 20 drones or males at a time. And she'll store that sperm in the sperm storage organ called the Spermetheka, and she'll use that sperm for the rest of her life. Now, if there is an issue with the drone, or the drone's producing low quality sperm, then the queen could be storing that low quality sperm, and that could contribute to a poor brood pattern as you observe as a beekeeper, or some of these brood issues that you would observe. And so for me, one thing I wanted to look at was, how do drones contribute to queen failure, right? What traits are important for a drone, and is drones an issue, especially in the United States? And so one thing I want to look at is low versus high sperm quality and low versus high fertility, the what are these traits and how do they contribute to some of these queen issues or just queen quality in general. So first, one thing we observed in the United States is drones contribute to queen failure, and so this issue I really outlined in the beginning. And one thing we noticed, especially talking to beekeepers, not only in the United States but Canada, was that queens are an issue. But we're also learning that drones may directly contribute to queen failure. So a lot of research has really been focused on the queen aspect of these failures, but drones are extremely important. So one thing they did was this researcher Jeffrey Pettis. You may have heard his name. He's pretty world renowned scientist, but he was based in Belton, Maryland. I actually think he's the president of Colas, and so he does a lot of work in Europe and Germany. But he heard these same reports by beekeepers, and so he tested queens that were raised in the control in the ARS, which is the USDA government facility, and then he collected queens that were filling on the east coast of the United States, and then queens that were filling on the west coast of the United States. And then basically what he does, he extracted that sperm storage organ called the Spermetheka and measured, okay, what percentage of the sperm was alive versus dead. And so here is kind of marker, right? One thing we'd expect was that for quality queens, that the sperm that she's storing should be nearly 100% alive. And so for the control, we see that the queen is storing sperm that's about 90% alive. So it's very viable. It's really healthy. And it might be a reason why the queens are doing so well. But for these queens that were reported failing in the East Coast and West Coast, I mean, you can see clearly from here that the sperm viability is, 50%, where it should be 90%. Now think about it this way: if a queen is storing sperm that's, 50 to 60% dead, just think about how that could affect the colony health, right? It's going to affect her egg laying ability. It's going to impact colony health, and it's going to impact all these other colony level metrics. And so one thing we're seeing is that drones are extremely important and they directly contribute to queen quality and, at least in the United States, queen failure. But drones are also important for a lot of these other colony level traits that you as beekeepers really want or are looking for, from your queens. So we know that if a queen mates with certain males, right, these can impact the genetic composition of a colony. And so a queen, right, her genetics is definitely represented in a colony, but if a queen's mating with 16 to 20 males, those different, I guess, genetic subfamilies within the colony are going to be really important for some of these traits. And so, these are traits that we think about for colony success, such as mite resistance, viral resistance, productivity to different landscapes, right, such as being high honey producers in the US versus German, and then this overall colony performance. And so just think about that drones are not only important for queen quality, but also these other colony level traits, because 50% of the genetics of every single colony is due to the males. So what actually makes a successful drone? And so when I first endeavored on this PhD and really focusing on drones, the one thing that really came to mind was we don't know much about the biology of the drones. We don't know much about which traits are important for drones. And so one thing we really had to do was outline these. And so I wrote this in a review, outline these traits and try to determine, okay, what traits should we focus on and really what biological traits are important for a drone. And I can split these apart into these three major traits. One is, sperm production. Two is sexual maturity. And then three is mating behavior. And so this whole lifespan takes around 54 days from when a drone egg is laid to when a drone goes on these multiple mating flights. And so first, sperm production. This occurs during the first 24 days of development. And this is probably the most important aspect for a drone, because a drone can only produce sperm during a small 2 to 3 day window. And so if anything happens during that 2 to 3 day window, then drones can never produce sperm ever again. And so, like, for example, I'll talk about this a little bit later in the talk, but if there's some harsh chemicals applied to the drones during this stage, then this can cause irreparable damage to the drones where they can never produce sperm ever again. And so I'll talk about that a little bit more, but this is probably the most important part of the drone, is the sperm production stage during the first 24 days of development. And once drones emerge as adults, they actually spend the first 10 to 14 days in the middle of the brood box. And so you guys are all beekeepers. We have the brood box, and in the middle is where all the eggs are laid and all the nurse bees are, and then the outskirts is where the honey is stored. Now the drones go to this middle of the colony because they really depend on the workers for everything, right? They depend on them for care, depend on them for feed, and they depend on them for warmth and some of these other important environmental factors. And so for some of my research, I actually found that if you actually take young drones and you put them in an incubator to try to feed on themselves or take care of themselves, a high proportion dies, and that's because this stage is extremely important and they really depend on workers for nearly everything. Now, this stage is also important because this is when drones reach sexual maturity. And I'll talk about this a little bit more, but what's different about sexual maturity for honeybee drones, and it's different than really closely related bees or any closely related insects, is sexual maturity isn't tied to the initiation of sperm production. And so for most, I guess, organisms, sexual maturity is tied to, I guess, when a male begins to produce sperm. For honeybee drones, it's a little bit different. And so I'll explain that a little bit more, but their sexual maturity is really unique. And then finally is this mating behavior. So once drones reach sexual maturity, they're basically kicked from where the brood nest is, where the nurse bees are, to the outskirts of the colony where the honey is stored. And the reason they do that is because they're basically transitioning from a high protein diet, which is provided by the workers, to a high sugary diet, which then need to go on these multiple mating flights. And so once a drone reaches sexual maturity, then basically they either mate or they die trying. And drones will go on these multiple mating flights around 30 minutes to 40 minutes per flight and around 7 to 10 flights per day. And so they're really just going to these regions waiting for a queen to arrive and ready to mate with them. And these regions are known as drone congregation areas, or I'll refer to them as DCAs. So I'll explain that a little bit more. But throughout this talk, I want you to think about these three major points because I'll come back to them. Point one, sperm production during development. Point two is sexual maturity during really early adulthood. And then three is this mating behavior. And all these three traits are important for drone success and drone quality. Okay, so first I want to dive a little bit deeper into sperm production during development. And so when an egg is laid, it's an unfertilized egg, right? From the honeybee male because they're haploid, whereas the females are diploid. And so the genetic structure of a male is already quite different. Now, during the egg, once the egg hatches into a small larva, and once you start beekeeping, I'm not sure when you call it begin producing brood, but it's when the brood is really small, the larvae are really small, and they're about the size of an egg. Now, this stage is important because this is when the testes are first produced during the drones. And so as the larvae get bigger and bigger and bigger to about 5 to 6 days after that young larva, the testes basically grow and get bigger and bigger. And then we get to the point where I guess we refer to this as a fifth instar larva. And so for a drone, this is, when you colonies produce drones, you can really tell whether a drone is in this stage because they're large. It almost seems like they actually don't fit in the cell, and they're in these kind of larger cells. And so if you're not familiar with I guess what a drone cell is and drone larva, you can find a few pictures online, but drones are raised in these distinct cells, because they're quite large. But during this larva development, this is when the testes are first produced. Now, once drones enter this fifth larva instar, which is when the drones are really large and almost seems like they don't fit in the cell, this is what we call spermatogenesis stage number 1. So this is the first stage of sperm production. Now, a few days later, the larva becomes upright, the cell becomes capped by the workers, and we call this a prepupa stage. And we call this because it's kind of a cocoon spinning stage. And so it's similar to, I guess, butterflies, where they go from a caterpillar to a cocoon spinning stage to kind of final form. And so honeybee drones do the exact same thing. And so once they reach this prepupa stage, this is the second stage of spermatogenesis. We call it number 2. And so when we kind of take a step back and look at when sperm is actually produced, it's only produced during this 2 to 3 day window, during this fifth instar larva and this prepupa stage. And after this age or after this stage, drones can never produce sperm ever again. And if anything happens during this stage, it can truly cause irreparable damage to the drone sperm and their fertility altogether. Now, once they pass through this cocoon spinning stage, and they enter pupation, which pupation is basically when all the tissues are reorganized and you can start to see the fully formed, I guess, body structure of a drone. And so from here, you can see the eyes finally, you can see the thorax, and you can see the abdomen, but also you can see the fully formed reproductive tract. Now, this stage is also important because this is actually when the sperm tails are formed. And so, and the sperm tail is important for swimming and fertility altogether. And so, when I talk about sperm is only produced in development, just think about sperm is produced in this 2 to 3 days during larval stage, and then the sperm tails are formed when they enter pupation. Okay, then point number 2 is this sexual maturity. So like I said, sexual maturity isn't tied to the initiation of sperm production because, once drones emerge as adults, they already have all the sperm they need for the rest of their lives. But sexual maturity is tied to actually the sperm physically swimming from the testes, which I have right here, all the way into the seminal vesicles. And so even though the drones have already produced the sperm actually has to swim to the seminal vesicles in order to be used during mating. And so this is one reason why they stay in the middle of the brood box and receive constant care by the workers, because the workers are really trying to help the drone through this process by feeding them, providing them warmth, and workers actually can tell whether a drone needs more care or not. They actually give it vibrational signals and actually give drones more care through feeding and tropholaylax if they do need this care. Now, one way to detect whether a drone is mature or immature is simply by, I guess, popping or squeezing the drones. And so you can take a chance, you can try this if your drones are producing colonies or when they produce colonies. Just take the drone and you just squeeze them by the thorax, and this endophallus, this mating structure will pop out. And you can see it right here. And so there, if you're not really sure what I meant, you can find a few videos about this. But you can just pop a drone and you can tell whether they're mature or immature. Now, this bottom picture is a picture of an immature drone. You can see that the endophallus is clear and there's no, I guess, brown or white fluids at the tip of the endophallus. And so this would be immature drone. And so this means that the sperm is still stored in the testes and not quite the seminal vesicles. And here's a picture of a mature drone where you pop them and you can see, this isn't the best picture, but you can see there's a brown sperm at the tip of the endophallus. And so this basically means that the sperm is already fully swimmed to the seminal vesicle and it's available during mating. And so this is one way we can track whether a drone is immature or mature. Okay, so finally, once a drone reaches sexual maturity, the workers are done taking care of them and they kind of force them to feed and care for themselves because the workers are really busy and they probably have more drones to take care of. And so once drones are kicked to the outskirts of the colony, or the honey supers if you have that, they'll start feeding on sort of a high sugary diet. And they do this because they need sugar as a resource in order to go on these multiple mating flights. And so what drones do is they go to these regions called drone congregation areas. And these areas can have 1,000 to 25,000 drones at a time. And so they're highly populated with these drones. And usually these DCAs are in the same region year in and year out. And so here in the United States, we know of some DCAs, and this actually has been confirmed in other regions of the world, including Australia, Africa, and probably in Germany also. But if there's a DCA, you can come back 40 years later and it'll still be the same area. And it's because the drones use these kind of landscape cues to choose these DCAs. And I'll talk about that a little bit more about how they choose these DCAs and why they're the same year in, year out. But they use these kind of visual cues to find these DCAs. And basically they just wait for a queen to arrive, and then they come back refueled and go back to these DCAs once again. And then once a queen arrives, these drones form these massive comets, I guess, these clusters, and they chase after the queen. And the queen will mate with 10 to 15 drones during these multiple mating flights. And so when we think about what causes a poor drone, or I guess we can frame it this year, what traits are important for drone quality, we have these really 3 major points: sperm quality, sexual maturity, and mating behavior. Okay, so what can actually cause a poor drone? So the first are these environmental factors, right? So I outlined 5 environmental factors I think are really important and things that can actually contribute to high versus low quality drones. The first is poor nutrition. And so if a colony is, foraging during a pollen dearth or the right nutrients aren't available, whether that's in a monoculture or whether that is a dearth, this can lead to the production of poor drones. Because, one thing about drones is the colony will only invest into drones if basically they have all the necessary nutrients to support colony health and survival. And so once they have sort of extra nutrients, they start investing more into drones. But if a colony suddenly enters a pollen dearth or into a location with poor nutrients around, this kind of forces the workers to invest less into drones. And so, we know that if we force a colony to pollen dearth, whether that's through pollen traps or whether that's in a monoculture, which I guess monoculture just means like one flower source, we know that drones are lower quality altogether. Second are these pests, pathogens, and diseases, and so we know these as Varroa destructor, viruses. There's several viruses associated with colony health, and then bacterial and brood diseases such as European foul brood and American foul brood. And so we know that these can really impact drone health. And one thing to keep in mind with this too is, if anything happens during drone development, which is when sperm is produced, such as the drones are the Varroa are feeding in the drone fat body, or the drones have viruses, or they have some kind of brood disease, then this can cause irreparable damage to the drones. The third of these miticides, and so these are mite treatments. And so I'm not positive which mite treatments you use in Germany, but I'm sure they're very similar. We know that the synthetic miticides cause probably the harshest impact on drones, and so these include amitraz, I guess fluvalinate, comofos. I'm not sure what type of treatments you guys still use in Germany, but these synthetic miticides can cause really harsh damage to drones. But even some of the organic miticides can impact drones. And so in the US, we think of these as sort of formic acid, oxalic acid, and also thymol treatments. So, their impact on drones isn't as harsh, but they can still impact drones to some extent. And so just think about some of these miticides when you think about drone quality and how they impact drones. For our pesticides, fungicides, and so in the United States, we have a high prevalence of neonicotinoids, which guess aren't used in Europe, but there's other fungicides and other pesticides that are used that can impact drones. And then finally, temperature. We know that temperature has a big impact on drones, and so these are probably the 5 major things I think are important for drone quality from the environmental part of it, but there's probably other reasons. Okay, finally are the genetic factors. And so when we think about a trait, it's not always environmental. There's also these genetic components. And so one way I really like to explain genetics is through sort of this random distribution. And so, for example, let's say we sample drones from 100 colonies from different beekeepers. What we'll typically get is this random distribution where a small set of the drones from these colonies will have low sperm quality, a small subset will have high sperm quality, and then most will be sort of right in the middle. Now, this can be due to environmental, but there could also be certain genes that could cause a drone to produce high quality sperm or a drone to produce low quality sperm. And so, for example, these colonies could have gene A, and that allows them to produce high quality drones, whereas these colonies have gene C that forces them to produce low quality sperm. And so genes are also really important, and something I really want to focus on throughout the rest of my talk, because one thing we think about drone quality is, okay, if drones are an issue and if they're low quality, especially in the United States, how can we use information to help beekeepers breed for better drones? And so one thing we can do is identify these genes. And so, for example, if we can identify, gene A is associated with high sperm quality, we can use this to help beekeepers select this trait. And so instead of selecting for, these drone traits, we can use it to select for, gene A through like a genetic screening tool. And so I'll talk about that a little bit more, but my focus is really going to be on the genetic part of it, because we do know a lot about the environmental factors that impact drones, but we don't know much about the genetic factors. Okay, so I'm going to talk about these 3 major points separately. So first, I'm going to be focused on sperm quality and really what genetic factors impact sperm quality, how much do these drones vary, and how can we use some of this information to improve drone quality in at least the United States. Okay, so we covered a lot of information, so I just want to kind of summarize this once again. Like, what is point one? So point number one is sperm quality, and so this occurs during the first 24 days of development, and sperm is produced during the 2 to 3 day window during this late larval instar when the drone larvae are really big and almost overflowing, and then also this prepupa stage when the cell is capped and they go under this kind of tissue reorganization. So this is when the sperm cells are actually first produced. And then spermiogenesis is when the sperm tail is formed. And so, this is probably the most important stage for drone quality, sperm quality, and fertility altogether. And so once they emerge as adults, they have all the sperm they need during development. So one of my first questions I had when I started my PhD was, okay, first, how much variation is there in these drone traits, right? So what are the sperm traits that are important, and how much do they vary across, honeybee stocks, breeds, lines, whatever you call them in Germany, across the United States? Two, okay, what is the genetic component of this, right? Is this variation driven by genetics or is it driven by environment? Because if it's genetics, we can identify these genes and use it in a breeding program. And so one thing we did was what we call a common garden experiment. And so as a geneticist, we really hate the environment because it makes identifying these genes really messy and complicated. And so one thing we do is we get these, I guess, different types of honeybee colonies and raise them in one environment. And that allows us to control the environment to the best of our ability. And so one thing we did, we started with, I have listed here 4, but we actually started with, 10 distinct lines in the United States. Now, in the United States, we have, honeybees are non-native to the U.S., and so what we have is, I guess, a bunch of mutts, and these are bees that are mixtures of bees from Africa, Europe, and Asia, basically, and so they're kind of a mixture of them. In the southern United States, we have a lot more bees that have a higher proportion of Africanized genes from Africa because there's like a sort of a zone where the Africanized bees didn't really spread to, but if you go to like southern part, like near Mexico, then these bees tend to have more Africanized genes. And so one of the lines we chose bees from there because we knew that they'd have more Africanized genes. In the western part of the United States, where California is, it's definitely more of a mixture of these European lineages, like Carniolan and Italian. So they have less, especially in the northern part of California, they have less of these Africanized genes and more of these Carniolan and Italian genes. And then we chose them from specific breeding programs. And so in the US, we have this breeding program for rural sensitive hygiene, which we call the pole line. And so I think there's some VSH breeding programs in Germany or at least in Europe, but it's very similar. We're breeding for the VSH trait. In Purdue and Indiana, where I did my PhD, we bred for what we call mite biting behavior. And so it's sort of a different form of hygienic behavior where the bees can actually sense the mites and they chew their legs off and then chew their exoskeleton. And so we call that a Purdue mite biting program. And so yeah, so I'm not going to explain them all, but one thing we did was we started with these 10 and we chose them based on the beekeeper, which beekeepers are commonly buying, whether they're part of a breeding program, or whether they have differences in their genetics. And we found that 4 lines display the most variation in these sperm traits. And so I'll be focusing on line 1 through 4, but I just want to really reiterate that we started with 10, but we dwindled down to 4. And then we raised them in one common environment. One thing we want to do was control for all the different environmental factors. And so once drones emerge as adults, we put them into one common colony. So they were raised by the same workers, they receive the same mite treatments, and they receive the same care altogether. And then we sampled drones when they were ages 10, 14, and 20, because what's interesting about drones is they go what we call a reproductive senescence. And senescence is just a really fancy way of saying aging. So drone sperm quality is typically low, around 10 days, because this is when they're still reaching sexual maturity, and so the sperm is still migrating and still really maturing. Once drones reach around 14 days or 2 weeks, this is when drone fertility is really maximized. And so, if you sample drones that are around 14 days, this is really their peak fertility. And then just due to natural aging, this sperm quality and fertility decreases over time. And so one thing we want to do was collect drones from these 4 different stocks and then measure the drone traits at 10, 14, and 20 in order to capture this whole senescence. And then we measured a wide range of sperm traits. And so we looked at what percentage of sperm were alive that drones were producing, how fast were they swimming. We looked at other sperm quality metrics. But one thing we did find was just the pure number of sperm that the drones were producing was highly correlated to all these other traits. And so I'll really be focusing on the number of sperm drones produced, but just remember it's correlated to what percentage of sperm was alive versus dead, or how fast the sperm was swimming, or some of these other fertility metrics we think of. But for now, just for the case of this presentation, I'll focus just on sperm number. Okay, so our first result was looking at sperm number and how they vary for these 4 distinct lines. And so right here I have line 1, 2, 3, and 4. And then on the y-axis, I have sperm number. And so this is a million sperm cells. And so typically, most of drones produce nearly 4 million sperm cells. But I think the top was 12 to 15 million sperm cells. And so it really ranges from 0 to 15 million sperm cells. Now, I highlighted here in green the 2 lines that show the most differences. And so I want you to focus on line 1 versus line 4. Now, the first thing, I guess my biggest takeaway here is that they do have this senescence where sperm quality is low at 10, maximizes increases to 14, and then decrease at 20. And so you see the senescence or aging, I guess, for both line 1 and 4. Now, what was interesting was we look at, I guess, peak fertility for drones from line 1 and 4. At age 14, drones produce around 6 million sperm cells. But for line 4, it's only around 4 million sperm cells. And so even though these drones are raised in the exact same environment, receive the exact same care, and everything is treated exactly the same. There's always going to be some environmental things we can't take account of, but we try to control that to the best of our ability. We found that drones from line 1 produce nearly 3 times more sperm than line 4. And so this suggests that there's this tremendous variation in these drone traits across the United States. Okay, so first we found that there's variation. That's really our first question. Next was, okay, this seems to be genetic, right? We did control the environment, so what's driving this variation might be genetic rather than the environment. And so one thing we did was we took what is known as the heritability estimate. And so this heritability estimate ranges from 0 all the way to 1. Now, a 1 heritability would mean that this trait is determined really solely by genetics. And so an example of this would be eye color, where we know certain genes are associated with high blue eyes, green eyes, or brown eyes. Whereas a heritability of 0 would mean that this trait is determined solely by the environment. And so there's not many traits like that. But we found that 33% had a 33% heritability for the sperm number trait. Now, while this doesn't seem very high because, right, still 70% of this is due to the environment. But this is actually really high for a trait like this. So when you compare this to most livestock or other industries, basically, heritability for sperm is typically around a 0.15 or 0.2. And so this heritability is actually really high. And so this suggests that not only is there variation, but there's genetics or certain genes causing the production of high versus low sperm. Okay, so we don't want to stop there. So we found that, okay, there's variation and this variation is heritable. Next, we want to find out, okay, what specific genes are causing this variation? And so one thing we did was we collected, I guess, this whole reproductive tract, which I'm circling right here, from about 250 drones. And we basically did the sequencing because you want to look at, okay, which genes are expressed and which gene expressions are associated with high versus low quality sperm. And so I'm going to explain all the methods because I think it's not as important for you as a beekeeper. But we found that there's 1,240 genes associated with sperm production in honeybee males. Now, these genes don't tend to behave the same way. They're typically in this cluster where on the outskirts of this cluster, we can see from these gray dots, these genes usually have an effect on a trait, but they usually have a really small effect on a trait. But there's usually a few genes right in the middle, and I colored it in orange, and these genes are usually really important for the trait of interest because they're not only driving the trait, but they're also regulating all these other genes. And so the one thing we were really interested in was pulling out this gene and seeing, okay, what the heck is this gene, and can we determine what's actually doing? Okay, so first we pulled up this gene, and we call it's called dynein. And first we found that dynein expression was significantly higher for drones that produce high amounts of sperm compared to drones that were producing low amounts of sperm. And so basically this confirms that dynein expression is higher for drones that I guess are highly fertile. Now dynein is basically the sperm motor protein. And so one way to think about this is you think about sort of a boat, right? A boat has sort of the main boat structure. You have the boat motor, and then you have the rudder. Sperm is similar like that, where the head is similar to the boat structure. The dynein is basically the boat motor, and the tail is the rudder. Now, what we found was that drones that produce low amounts of sperm, their dynein or their boat motor just wasn't working properly. And so what this means is the sperm structure is actually there, but for the drones that are producing less sperm or lower quality sperm, the motor just isn't working properly. And so this seems to be one major driver for low sperm quality and low sperm production for honeybee males. Okay, so the first part of the talk was really focused on the sperm quality traits. Next, we're getting to point number 2, and this is sexual maturity. Now, once again, sexual maturity for honeybee drones or males is tied to the sperm swimming from the testes, which is right here, into some of the vesicles. Now, the one thing we found from the previous, I guess, part of the talk was, okay, the drones are producing less sperm. The sperm swimming just isn't working properly. It's probably because the sperm motor just isn't functioning. And so if that sperm isn't able to swim properly, or something's impacting the sperm, then this could actually impact their sexual maturity. And so, for example, let's say a drone is going through development and produces, 4 million sperm cells, but only, 1 million sperm cells are actually able to swim and move to some of the vesicles. I mean, even though the drone produces 4 million sperm cells, if only 1 million can actually swim, then that's all the drone can actually be used during development. But let's say the sperm doesn't swim at all. That could actually mean that these drones never reach sexual maturity because the sperm is never able to swim here. And so that could actually cause the production of what we call dud drones, or drones that are unable to produce sperm. And so this is kind of an example of a dud drone, right? It can be immature, or it can also be a dud drone, where if you pop a drone and there's no sperm at the tip of the endophallus, we call these duds. Whereas a mature drone, where the sperm is actually physically swimming, we can pop them and we can see that there's actually brown sperm at the tip of the endophallus. And so this would suggest that the sperm is swimming properly and these drones are mature. And so our next question was, okay, these drones are producing less sperm. Is that actually impacting the number of drones that reach sexual maturity or not? And so we did the exact same experimental design with our 4 same lines, and I'll once again call them line 1, 2, 3, and 4, and we raised in the common garden environment. But this time, instead of looking at sperm traits, we basically popped the drones at ages 10, 14, and 20 to capture this, I guess, drone sperm aging. And we measured, okay, what proportion of drones were mature at 10, 14, and 20? And like I said, the one way we measured sexual maturity was simply popping the drones and seeing if they were actually producing sperm. And so our next question was, how much do these sexual traits vary, and is there a genetic component to this variation? And so I'm focusing on, oh, sorry about that. So I'm focusing on age 20 because, I'm going to focus on age 20 because our expectation was, okay, most of the drones are probably not mature at age 10 because the sperm is still swimming and they're still kind of reaching sexual maturity. By age 14, most of the drones should reach sexual maturity because this is when their fertility is really maximized. But for age 20, we expect that really all drones should be sexually mature by age 20. And if they're not sexually mature by age 20, I mean, it's definitely possible that these drones really may never reach sexual maturity because age 20 is really late. And so I'll be focusing on age 20 because what we expect is most of the drones, nearly 100%, should be mature by at least age 20. And so here are results. So this is the percentage of sexually mature drones for our lines 1, 2, 3, and 4, which are the exact same lines that I talked about previously and kind of explained through our common garden experiment. Now, so for line 1, which is also a line that produced the highest quality sperm and the most sperm, about 75% of these drones were mature at age 20. Only 50% of the drones were mature for line 2 and 3 age 20, but then a staggering only 33% of the drones were sexually mature at age 20 for line 4. Now, what's really crazy about this is, one, these drones were raised in the exact same environment, and so it's really interesting that drones from line 1 are producing just a high proportion of sexually mature drones. But two is that 70% of the drones aren't sexually mature by age 20 for line 4. And so what this means is kind of from a broad perspective for beekeepers, at least in the United States, is that beekeepers are producing a bunch of drones, and it's possible that these drones that aren't mature by age 20 may never reach sexual maturity. And so that means that only 33% of the drones in the population are really able to mate and pass their genetics to the next generation. And so this is really concerning in a few different reasons, and we're kind of assessing this a bit more. So this data is still a bit preliminary. So next, okay, we see that there's a tremendous amount of variation between these lines, especially between line 1 and line 4. Next, we want to look at the heritability estimates. So how much were genetics playing a role in what we were seeing? And so I don't have this up here, but we found that the heritability was about a 0.4, which is really high and significantly higher than the sperm heritability. And so we see that genetics are playing a huge impact on these traits. Okay, so we didn't want to stop there. So we see that there's a lot of variation, and it seems to be determined by genetics. Next, we want to do the exact same thing with sperm, but with sexual maturity. What genes are causing immature versus mature drones? And so what was interesting was we found that genes expressed in the brain was actually correlated or associated with sexual maturity rather than in the reproductive tract. And this sort of makes sense conceptually, because in order for a drone to begin these multiple mating these mating flights, they have to go through sort of the behavioral changes in the brain that really kind of gears them up or prepares them for these mating flights. And so what we're seeing is that sexual maturity might be tied to the behavioral changes in the brain that kind of initiate the drones to fly and go on these mating flights. Now, in total, we found 825 genes associated with sexual maturity in the brain. And like I said, they're in these clusters, and we found that there's one gene in the middle of the cluster that seems to be highly correlated or associated with sexual maturity. And so we pulled out this orange gene to see, okay, what the heck is it doing? And so first off, we found that drones that are immature have lower expression of this gene than drones that are mature. So it seems to have some functionality in sexual maturity. But next we found that this gene is called RAS32. And so RAS32 is sort of like it's sort of like the truck driver, or the mail carrier, whatever the German equivalent of that is, for the sperm. So what it does is this truck driver, it collects the nutrients, it collects cargo, it collects everything the sperm needs, and actually physically drives it to the sperm motor protein dynein, which I talked about earlier, and gives those nutrients to the dynein in order for it to swim. And so whether it's transporting spark plugs or gasoline or petrol oil or anything like that, this truck driver is physically carrying those nutrients to the sperm. And so what could be happening while these drones aren't reaching sexual maturity or they're producing low amounts of sperm, especially for line 4, is something's happening at the truck driver level where these nutrients or these cargo aren't being transported to sperm. And so I'll explain this system a bit more next, because so I guess your next question is, okay, if these truck drivers are being produced in the brain and this dynein is in the sperm, the question is, okay, how does these truck drivers get all the way down to the sperm? And so one thing we did was we found shared genes, or genes that are shared in the brain and the testes that could be allowing this truck driver to get to the sperm. And so we pulled out the gene right here to see what it was doing. Okay, so we found this gene was called BCAATL-D. And BCAATL-D is sort of the highway or the road system or the sidewalk, or whatever you call it. It's basically this road that allows these truck drivers to swim all the way to the sperm. Now, these are probably the three most important genes of RAS32, the Brcado D, and the dianine. And you have to remember these specific genes because it's probably less important for you as a beekeeper. But the one thing I want you to take away from this is, okay, line one drones are producing, line one colonies are producing higher quality drones, whether that's sexual maturity, whether that's sperm quality. And the reason they are is because something's happening at one of these three levels. Whether that's something happening at the truck driver level, where there's a lack of truck drivers, there's a lack of cargo and nutrients from the transport. Something could be happening at the road system way, or the highway, where maybe there's potholes or there's road construction where the truck drivers aren't physically able to drive. Or there's something at the sperm level where something's actually physically wrong with the dianine protein. And so, something's happening at one of these three levels, really preventing high sperm quality for these specific lines. And so one thing, I guess, for me that I want to do next is, okay, I found these really important genes. How can we, I guess, screen colonies? So do genetic testing on colonies to help beekeepers identify these genes, and so we can breed for improved drone quality. Okay, so the first part talk was really focused on fertility and some of these sperm quality traits. But a drone success isn't really reduced down to just the sperm level. It also is important for their mating behavior, because a drone could produce really high quality sperm, but if their mating is hindered or something's wrong with their mating, they still can't pass the genetics or the sperm to the next generation. And so point 3 is really focused on some of this mating behavior. And so just kind of a little bit overview on this mating behavior, and I think I have a video on here I'll show you, but drones go through these areas called drone congregation areas, which are populated by 1,000 to 25,000 drones, and these are the same DCAs year in, year out. And they go to they locate these regions based on these kind of landscape cues, whether it's a forest edge next to a tall building or on the bottom end of a valley. They use these kind of visual cues to find these DCAs. And basically, they kind of wait in these DCAs for a queen to arrive, because queens use very similar visual cues to find these DCAs. And so once a queen arrives, the drones will basically form this comet and then chase after the queen. And either they'll mate or they'll just try again until they die trying. And so let's make this video to play quick. And there's a lot of these videos online. If you go to YouTube, you can search them. This is just one I found. And so this beekeeper actually had a stick, and at the end of this really long stick, they put a virgin queen, or maybe put like a queen pheromone, something to attract the drones. And as you can see, all these drones are kind of flying around this queen. So you can see there's probably thousands of drones right here. And so, back here. So there's about thousands of drones right there. It's basically, there's some better videos, but the drones actually kind of knock each other out of the way, and they're kind of fighting for access to the queen. Now, once a drone kind of jumps on the back of the queen and starts mating with the queen, what happens is they both become paralyzed or unable to move, and they'll actually fall, 3 to 4 meters down the air, and they're just unable to fly because they're going through this mating process. Once the insemination is finished, the whole reproductive tract and the testes and the endophallus for the drone is actually ripped out of the drone's body, and the drone falls and dies. And so it's a very vicious process, but drones can only mate once, and so by doing this, it probably allows them to inseminate the queen with more sperm. And so there's some really cool videos on this, so I really encourage you to look at some of these videos. Okay, so this was a study done in Puerto Rico where they were trying to track where the drones are going, where the DCAs were, and really the drone mating behavior. And so this is what this region in Puerto Rico looks like. And so from all these triangles, you can see these are the actual colony locations. And so you can see right here, there's probably like 30 to 40 different colony locations that they studied. And then what they did was they put this antenna on the back of their drones. And by having like 3 to 4 people, they were able to triangulate and figure out where the drones are going. And they found that even though there's about 30 of these, colony locations, the drones are only going to about 4 to 5 different drone congregation areas, which are in these small blue squares. And what was really interesting, what they actually found was that the drones were actually, when they first started flying, they go to 3 to 4 of these DCAs. But once they get older and mating becomes more important because they don't have much time left in their life or their sperm quality is being depleted, they tend to go back and forth between one of these DCAs. And like I said, these DCAs, and how they chose them was really unique. So they're basically able to correlate the DCAs to some of these kind of landscape markers. And so they found that they're usually located near a forest edge, in the bottom of a valley, or next to sort of a large structure, such as like a football stadium. Okay, so next was, we looked at sperm traits. Next is, how much do these flight traits vary? So we have 4 different lines. Next, we put these small RFID tags on the back of the drones. Now, these aren't GPS trackers, right? We can't tell exactly where the drones are going, but it tells us when a drone is leaving or coming back from the colony. And this gives us a lot of really important information about when the drones first begin to fly, how many flights per day, how long are these flights, how long are they refueling within the colony, all these really important flight traits. It's not necessarily mating behavior, but it still gives us a lot of information about some of this flight behavior. And here's an example of a single drone that we tracked. So you can see for the first three days, the drone isn't flying at all. Around 4 to 5 days, the drone begins flying around 20 to 30 minutes per flight. And then you see right here, it actually dropped because it's raining that day. So, there's definitely no flight information because there's a heavy pour. And then next day, they started flying again. And so you can see the type of cool information we get from this. Okay, so next we looked at, how much do these flight traits vary by line? And so first we looked at age of first flight. You see for drones from line 1, their first flight age was around 8. For line 2, 3, and 4, it was around 6. And so 2 days is definitely massive for life of the drone. And so what's interesting is, why are these drones actually delaying their first flight by 2 extra days? And one reason is, by delaying flight by an additional 2 days, it allows them to receive more care by the workers. It allows them to improve their sperm quality. And so one reason that line 1 may actually produce more sperm and high quality sperm is because they're actually delaying flight by an additional 2 days. Because there's definitely trade-offs between initiating flight and then also the sperm traits. Next are flights per day. So line 1 drones took around 10 flights per day, to 8 flights per day, and then 3 and 4 or 6 flights per day. And so you see there's a lot of variation in number of flights. But also interesting for line 1 was these drones took, an additional 4 flights per day. And so that could also be correlated to them actually delaying their age of first flight. And so, I want to kind of take a step back and kind of summarize. So, one of our major questions was, how much variation are there in these traits? As we see that there's a lot of variation in sperm quality, sexual maturity, and then mating behavior, which are our 3 major points. We see that all these traits are highly heritable, so the genetic component. We found that there's about 3 to 4 core genes associated with this variation, and so they seem to be really important for these traits. Now, what does this actually mean? The next is, how does this actually contribute to queen failure and queen quality altogether? And so I'll really focus on the beginning of the talk is, how important are drones and how do they contribute to some of these queen traits? And so the next question is, we have these lines and these drones vary in these traits, but next is actually inseminating queens and figuring out how this impacts queen fertility and queen traits. Next, the next thing too is, because this is based on location, so performance is based on location. So we did this common garden experiment in central Indiana, in the central United States. And so if we repeated the study in different regions of the US or different parts of the world, they may actually perform differently because, line 1 this may perform really well in our environment. But if we repeated the study in Germany or repeated it in other parts of the United States, line 4, which did worse in our study, may actually do better. And so one thing we need to do is actually repeat this in multiple environments to see this genetic by environmental interaction, because we can't always reduce everything down to genetics because the environment plays a really, really important role. So that's kind of the next step, is looking more at the environment. And then next, the next thing you may think about, why are these lines, especially line 4, producing such low quality drones? Wouldn't that be selected out of the population? And one thing I've really been thinking about is when you breed for some of these production traits, such as honey production, or breed for maybe rural defense behavior. When you breed for that, that tends to have a negative cost on fertility. And so this example in dairy cattle in the United States, they've been breeding for high milk production for the past 6 years. And you see that milk production for each cow has dramatically increased over the 6 years. But the fertility and pregnancy rate have also drastically increased. And so usually these traits are interconnected. And so one thing we could be seeing is if beekeepers are selecting for, honey production or rural defense or some of these other. And so with that, I will take any questions. I want to thank all my funding partners, including some organizations in the United States, such as ABF, Project 8%, and then the North Dakota Beekeeper Association, and then my advisor, then help. And if you have any more questions, I know the translation might be a bit iffy, so hopefully this works out really well. But if you have any additional questions, you can feel free to email me. And with that, I will take any questions.
Okay. I'll have to prepare some slides. I'll do some rearrangement.
Okay. I can move up the slide too if you need me to.
So we can watch the slides.
Cool.
And place the questions. For some reason, I have my echo online. So, okay, now it's gone. So now we can do the Q&A session. I'll have to go back through the questions because
Sure.
I can't protocol the questions. The first one, a very practical question. If we test the quality of one drone of a colony or a group of drones, can we extrapolate to the quality of other drones on his brothers?
Sure. That's a really good question. And so the first thing about the genetics of a drone is the queen lays all haploid eggs, and so they're unfertilized eggs. And so the drones, especially if they're from that same queen, are highly related to each other. And so I don't know the exact number of drones you would need to test, but if you test 30 drones, I think that'd be a good number. You can really predict the quality of drones throughout the entire colony. And then just kind of come from a practical beekeeper perspective, if you're interested in maybe testing or looking at some of these drone traits, some of these tests, they do require some equipment and some training. But one thing you can do, and this is something I recommend to beekeepers, is you can simply pop the drones and just look at the percentage of drones that are mature versus immature, and that's sort of a quick and easy way for a beekeeper to select on that. You can also look at drones and how much, if you're not familiar with this, but if you pop a drone, you can tell that some drones have more or less sperm, and as you get more experience, you can really tell the differences. And so you actually pop the drones and see, okay, which drones are producing more or less sperm, and then kind of choose based on that.
Also als Antwort gibt man, da Drohnen eines Bienenvolkes eng miteinander verwandt sind, ist schon damit zu rechnen, dass es eine Korrelation gibt. Wie viele Drohnen man jetzt tatsächlich testen muss, weiß er nicht. Aber als ersten Versuch, den man auch als Imker am Bienenstand, am Bienenvolk tun kann, schlägt er vor, dass man Drohnen zum Stülpen bringt und einfach die Quote ermittelt, wie viele Drohnen sind Stülpen, zeigen ein ausgereiftes Geschlechtsorgan, wie viele Drohnen stülpen nicht oder zeigen diesen durchsichtigen, wässrigen Phallus. The next question. Can artificial insemination cause to select for weak sperm quality or weak drone flying ability? Because drones who have been used for artificial insemination never had to fly and conquer for a queen?
Sure, sure. That's a really good question because I guess yeah. So that's a really good question because if you're selecting the drones for insemination, right? Those might not be the strongest or fittest drones at all, and so I guess you could be basically selecting for lower quality drones. Now with insemination, right, your goals are a little bit differently, where you're looking at some of these other traits. But because of that, you could be basically selecting for drones that produce lower quality sperm or lower fertility altogether. And so one thing that because I do tell beekeepers that are doing insemination if they want to kind of look at some of these drone traits before inseminating queens with drones is make sure that they produce, at least 2 microliters of sperm. And if you're doing insemination, you can tell how much 2 microliters is, and that's one way to kind of select for higher quality drones. But kind of back to your question, we don't know if they're the fittest flyers or if the sperm is actually higher or low quality. Just kind of doing a visual cue. So yeah, that's definitely one consideration. It could be leading to what we're seeing.
Also er hat geantwortet, dass die Sorge berechtigt ist, dass deswegen Imkern, die instrumentell besamen geraten wird, eine gewisse Mindestmenge von Sperma vorauszusetzen, bevor ein Drohnen für die Besamung eingesetzt wird. Ob das dann und in welcher Weise dann die Drohnenqualität optimiert ist, ob die jetzt fliegen können oder ob die gleichzeitig eine hohe Spermazahl haben und die Beweglichkeit der Spermien stimmt, lässt sich darüber natürlich noch nicht feststellen, aber das ist die einzige praktikable Möglichkeit, die man ohne großen Laboraufwand machen kann. Okay. I have to scroll a little. There was a question, I can't show it now, in the time where you talked about the age of the drones, 10, 14, 20 days. We sometimes see drones wintering. So we have drones in January or February, even April, before the next drone combs have been produced by the colonies. Would you expect that they are still viable?
Yeah. So first with experimental design, I stopped at age 20 just because I basically ran out of drones and I had a very specific time period. But I do know some drones that I tracked in previous experiments that lived for 70 to 90 days, and so these drones can live quite a bit longer than the 20-day period that I looked at. It's just that their sperm quality dramatically decreases after 20 days. Now as for drones overwintering, I mean I think that's a really interesting question. Typically a colony will probably overwinter with just a very subset of drones, and ostensibly maybe that's a way for them to keep these drones in the population, especially when they're able to fly and when the weather changes. And so it's possible that workers give them more care to maintain that sperm quality. It's possible, because the colony plays such an important role for some of these sperm traits that that they're probably helping this drone maintain sperm. But if it is past this 20 days, which would be the case for these overwintering drones, then it is probably likely that the sperm viability and sperm quality is going to be quite a bit lower. I wouldn't say they would be duds or it'd be 100% dead, but I think the sperm quality would be lower than if we sampled drones at age 14, for example.
Also ich finde es interessant, dass wir überwinternde Drohnen überhaupt finden. Und er geht davon aus, dass die Qualität dieser Drohnen marginal ist, dass sie im Prinzip unfruchtbar sein dürften. Untersucht hat das nicht, weil er einfach keine solch alten Drohnen zur Verfügung hatte. Next question. We discuss Varroa treatment by heat treatment. And there's the discussion that this heat treatment might impair the quality of drones. Do you know something about this?
Yes. So as for mite treatments, I guess we have different tiers of mite treatments, and I guess it seems like heat treatments are more common in Germany. It's become more common in the United States, but we do know that heat stress drones this produce lower quality and basically are unviable. There's some really cool studies in Canada showing that if we stress the drones even for a couple days, that the sperm quality would be practically inviable. And so I would guess that some of these heat treatments would be having a huge effect on drones. We even know if we get some really hot days here in the United States, like if it gets to 35 C or like quite a bit higher, that the drones will actually kind of fall from the air and die in the air because it's just quite a bit hotter. And so yeah, these treatments definitely would be impacting drones. Now, as I always want to reiterate this, because I get this question a lot, especially with miticides or some of these other treatments, is I'm not saying that you shouldn't use these treatments because Varroa are really the number one issue facing beekeepers across the country. So it's really important to use these treatments, even if it does have an impact on drones. But if you are planning on mating, which would happen, I guess, in the spring here, or whenever it happens in Germany, it might be a good time to use alternative treatments. So you are producing a high quality drone stocks for your queens to mate with or your neighboring queens to mate with. And then as the season goes on and your queens are fully mated and you want to worry about that, making sure to focus on the mite issue and controlling that's gonna be really, really important. And so I like drones are important, but make sure to time that out so you can still control for varroa and still produce high quality drones. So it's a difficult question, but something that you can time out.
Another question in this area of questions. The typical varroa treatment turnus in Germany stops at Christmas. And we don't start to treat before start of July.
Okay.
Would you expect a mite treatment done that late in the season to have impact on drone rearing in April or May?
Yeah. So yeah. That's a really big time span, and I guess it really depends on when your colonies begin producing drones. So I mean, if your colonies are producing drones before for that mite treatment, and it's like 50, let's say your colony are producing drones 50 days before that mite treatment, then your drones should be fine. I mean, there's always gonna be some residual things in the wax, but your drones should be fine. But if you are applying those mite treatments, and any mite treatment can impact drones, it's gonna impact the drone quality altogether. Now, the other consideration is, you could be doing things the right way, but your neighboring beekeeper uses a lot of treatments and impacting drones. Like your queen's still gonna mate with those, and so that adds a whole nother layer to this complexity because you can really control for your drone population, but you can't necessarily control for your neighbors' drones.
Okay, that's kind of a breeding selection program for my drones.
Exactly.
Is there a practical possibility to check for drone quality? For instance, are there anatomical cues to test for that a drone is viable and has good sperm quality without actually testing using a microscope or something to check for mobility of the sperms?
Sure. I would say the best way, because one thing we did find was that sperm number was highly correlated to all these sperm quality metrics. And so sperm number is probably one of the easiest things that you can look for. And you don't need a microscope to do this. You can look at it visually. And kind of what I was talking about when you pop a drone, and if you want more information, you can probably Google a mature versus immature drone. I actually sent you that picture. We have a really good picture of the drone sperm. And there's usually the brown sperm at the tip of the endophallus. And if you look at that, you can tell whether a drone's producing a lot or a little amount of sperm. And so, just visually, you can tell whether a drone's producing a half a microliter versus, 3 microliters. And so, if you wanted to, breed or look for colonies that do produce high quality drones, you could simply just pop the drones and just kind of visually look at the drones that are producing the highest amount of sperms versus the least amount of sperm. Or you can just measure the drones that are mature versus immature, right? Like if you're popping, 30 drones, one colony may have, I don't know, like 70% of the drones mature, whereas another colony might have 30%. And so it's one way to look at that also. And it's probably the more practical. Now, if you want to do a microscope, there's definitely ways to do it. Just requires the infrastructure and the training.
Okay, one last question for today. And I think it's an important question. What can the beekeeper do to aid in drone quality?
Yeah, so I guess one thing is, so I can't speak about this in the United States because typically we have a spring, and that's when beekeepers are buying queens or producing queens, and this is when the queens are actively going on these mating flights. And then after that, I mean, they're storing the sperm for the rest of their lives. And so I really think this timing is really important. And so making sure the time your mite treatments, for example, is gonna be really important. So you're not impacting the drones not only for yourself but also your neighbors. And the last thing is, making sure to control your mite population. And so one of my first graphics I showed was, during I guess drone development, this is probably the most important stage for sperm quality and drone fertility. Now, if your colonies are just filled with mites, I mean, some personal experience. If you put like a drone ferment into a colony, they're basically mite magnets and they're full of mites. And so those mites are basically attracted to the drones. And so they not only feed on the fat body, which lowers the immune system and sperm of the drone, but they also transmit some of these deadly viruses. And so not only timing your mite treatments, but making sure your mites are low is probably the most important part. And if you have the infrastructure and you're willing to select for higher drone quality, you can definitely do it. I think there's a lot of practical ways you can implement this into a breeding program. So that's definitely another way to approach it.
Okay, thank you very much for aiding in this presentation. It was very interesting. Thank you for answering some of the questions. I will have to do a translation next week. Perhaps I will come back to you to check questions that arise during this work. So, damit sind wir auch schon für unsere deutschsprachigen Gäste am Ende der Sendung. Nächste Woche werde ich versuchen, das, was Garett uns heute mitgebracht hat, auf Deutsch zusammenzufassen. Ich habe mit ihm gerade abgesprochen, dass er mir vielleicht das eine oder andere Detail, was mir sicherlich auffällt, wenn ich diese Übersetzungsarbeit mache, noch erklären wird. Damit wisst ihr, was nächste Woche auf euch zukommt. Viel Spaß, schönen Abend noch. Wir sehen uns nächste Woche. Thank you very much, Garett. Have a nice day. I will stay in contact. And that's the end for this evening.
Well, thank you. Yeah, if you have more questions, just shoot me an email, and I'm happy to answer them and help you out. So, no, really enjoyed it.
Thank you very much.
All right. Bye.